Hey everyone, welcome back to the Spark Academy YouTube channel. In today's video, we're diving into the fascinating world of cell division, focusing on two key processes, mitosis and meiosis. These biological events are central to life itself. Mitosis helps organisms grow and repair tissues, while meiosis enables sexual reproduction and genetic diversity. We'll break down each phase of mitosis and meiosis in detail, explore their differences in a clear narrative form, and connect them to real-world biological examples. Section 1. Mitosis, the engine of growth and repair. Mitosis is the process by which a single somatic cell divides to produce two identical daughter cells. It's essential for the growth and development of multicellular organisms and for repairing tissues. Think of it this way. When a child grows taller or when you heal from a paper cut, that's mitosis in action. Before mitosis begins, the cell undergoes interphase, a preparatory stage where it grows, carries out normal functions, and duplicates its DNA. Once ready, the cell enters the mitotic phase, which includes prophase, metaphase, anaphase, telophase. Prophase, the chromosomes condense and become visible under a microscope. Each chromosome is made of two sister chromatids joined at the centromere. The nuclear envelope starts to dissolve, and spindle fibers begin to form from the centrosomes. Metaphase. Chromosomes line up along the metaphase plate, the cell's equator. Spindle fibers attach to the centromere of each chromosome from opposite poles of the cell. Anaphase. The centromeres split, and the spindle fibers pull the sister chromatids apart to opposite poles of the cell. Once separated, each chromatid is considered a full-fledged chromosome. Telophase. The chromosomes reach the poles, and new nuclear envelopes form around them. The chromosomes begin to uncoil and return to their less condensed chromatin state. Cytokinesis follows, where the cytoplasm divides, and the cell membrane pinches inward to form two genetically identical diploid daughter cells, each with the same number of chromosomes as the original cell, 46 in humans. Section 2. Real-world applications and relevance of mitosis. Mitosis is continuously at work throughout life. In humans, it regenerates skin cells, replaces red and white blood cells, and enables organ repair. It is the basis of asexual reproduction in some organisms, such as hydra and bacteria. Though bacteria use binary fission, the concept of clonal reproduction is similar. A failure in the regulation of mitosis can result in cancer. Cancerous cells are those that divide uncontrollably and can form tumors or spread throughout the body. Understanding mitosis helps scientists develop treatments that target cell division. Section 3. Meiosis, the source of genetic diversity. Meiosis, in contrast, is a two-part cell division process that produces gametes, sperm in males and eggs in females, with half the number of chromosomes haploid. In humans, meiosis reduces the chromosome number from 46 to 23, so that during fertilization, the resulting zygote restores the diploid number. Meiosis is divided into Meiosis I and Meiosis II. Meiosis I is the reductional division. Prophase I. This is the most complex and longest phase. Homologous chromosomes pair up in a process called synapsis to form tetrads. Then, crossing over occurs, a critical event where homologous chromosomes exchange genetic segments. This shuffling increases genetic diversity. The nuclear envelope disintegrates, and spindle fibers emerge. Metaphase I. Tetrads align at the metaphase plate. Each pair's orientation is random, contributing to independent assortment, another source of genetic variation. Anaphase I. Homologous chromosomes are pulled to opposite poles of the cell. Importantly, the sister chromatids remain together. Telophase I and cytokinesis. Two cells form, each with half the original number of chromosomes, haploid, though each chromosome still consists of two chromatids. Meiosis II resembles mitosis. Prophase II. Chromosomes condense again in each haploid cell, spindle fibers form, and the nuclear envelope breaks down. Metaphase II. Chromosomes align along the metaphase plate. Anaphase II. Sister chromatids are finally pulled apart toward opposite poles. Telophase II and cytokinesis. For genetically unique haploid cells are formed. In males, all four become sperm. In females, typically one becomes a functional egg, and the others form polar bodies. Section 4. Key differences between mitosis and meiosis. While mitosis and meiosis share similarities, such as chromosome duplication and the use of spindle fibers, their purposes and outcomes are fundamentally different. Mitosis occurs in somatic, body, cells, and results in two identical diploid cells that are clones of the parent cell. It supports growth, maintenance, and repair. Meiosis, on the other hand, occurs only in germ cells, cells destined to become gametes. It produces four non-identical haploid cells, ensuring that offspring inherit a mix of genetic traits for both parents. Genetic variation in meiosis arises from crossing over during Prophase I and independent assortment during Metaphase I. These mechanisms explain why siblings from the same parents can look and behave very differently. Mitosis does not promote genetic variation, which is why cells produced through mitosis are genetically identical to the original. Moreover, mitosis involves a single round of division, while meiosis includes two consecutive divisions. This is critical to ensure the chromosome number is halved in gametes, allowing for the restoration of the diploid state at fertilization. Section 5. Real-world implications of meiosis. The genetic reshuffling that meiosis provides is essential for evolution and adaptation. Without it, populations would lack the variability needed to survive changing environments. However, meiosis is not foolproof. Errors such as non-disjunction, where chromosomes fail to separate properly, can lead to genetic disorders. For example, if an extra copy of chromosome 21 is present due to non-disjunction, the individual will have Down syndrome. Meiosis is also exploited in agriculture and animal breeding. Selective breeding programs use the genetic diversity produced through meiosis to develop crops with better yield, resistance to disease, or desirable traits. Section 6. Conclusion and summary. To summarize, mitosis is the process of creating identical diploid cells, crucial for growth and healing. Meiosis is the process of producing unique haploid gametes, essential for sexual reproduction. Mitosis results in uniformity. Meiosis ensures variation. Each has distinct stages and outcomes, but both are vital to life. Understanding these processes not only deepens our grasp of biology, but also enhances our ability to develop medical treatments, improve agriculture, and explain human diversity. Thanks for watching. Be sure to subscribe and hit the bell icon for more in-depth biology content. Drop your questions and future video suggestions in the comments.